Additionally, there are some ducts from those accessory organs that are going to empty directly into the duodenum and release some other additional fluids that are going to assist with digestion of nutrients. So first we have the liver here. The liver is going to produce a solution called bile. So the liver produces bile and then it stores bile in the gallbladder so that the bile is all ready to go. The function of this bile is to help emulsify fat so that we can digest it more efficiently. We'll talk in more detail about that in just a second. And then there's also a duct from the pancreas that is going to secrete pancreatic juice into the duodenum. This pancreatic juice is produced by cells in the pancreas called acinar cells. I just want to take a quick moment and remind us about how amazing the pancreas really is. The pancreas has different types of cells that are responsible for producing hormones, glucagon and insulin, and then a separate set of cells that we're talking about here, the acinar cells, that are responsible for producing a whole cocktail of enzymes. The pancreas is going to produce many different kinds of enzymes that are specific for digestion of carbohydrates, another suite of enzymes specific for the digestion of lipids, and then again another set of enzymes that are specific for the digestion of proteins. So a whole cocktail of enzymes. The other component that is very important in the pancreatic juice is bicarbonate, or HCO3, because remember that the chyme that is entering from the stomach is very, very acidic. And in order for all of these enzymes that are coming from the pancreas, in order for those enzymes to be able to efficiently catalyze the reactions and break down those macronutrients, they have to be at a higher, less acidic pH. So the bicarbonate from the pancreas is very important in order to bring the pH up and make it less acidic so that those enzymes can function. That bicarbonate is going to be neutralizing the acidic chyme. So let's talk in a little bit more detail about bile. But first, let's think about why do we need bile in the first place? Like, what is the problem here? So I want you to think about salad dressing. I like to make salad dressing with olive oil and balsamic vinegar. And what I know is that when I make a salad dressing like that, I can shake it up and it'll get mixed. But then over time, they're going to separate out where you're going to have the olive oil, the lipid on the top, and then the heavier hydrophilic balsamic vinegar along the bottom. Now, a similar situation is going to happen in our gastrointestinal tract, where the lipid component of our diet is going to float to the top, and then the hydrophilic, the water-loving components are going to float to the bottom. Because so far, the gastric juice, that chyme, it's a very watery-based solution. So our lipids that we ate in our diet are just going to float to the top like unmixed salad dressing. And this is a problem because the enzymes from the pancreas need to be able to access the lipid in order to digest it and break it down, and it can't access the lipid if it's just floating along the top like unmixed salad dressing. So what we have to do is we have to take these big, huge lipid droplets that would be floating on the top of our very watery chyme, and we have to break them down into smaller lipid droplets so that we can increase the surface area to allow the lipases, the fat breaking down enzymes that come from the pancreas, so that those lipases can actually access the fat and digest it. So what we're going to do, so up here we have some pictures of these big, huge lipid droplets. And then we see that there's another component that gets added along the outside, and these are the bile salts. So bile salts are produced by the liver, and they have kind of some interesting chemical properties in that they have one side that is polar, so water-loving, hydrophilic, that's represented by the green dots. And then the other side of the bile salts are nonpolar or hydrophobic. So the hydrophobic nonpolar side to the bile salts will orient themselves towards the lipid droplets. And then the polar side, hydrophilic side of the bile salts will orient themselves towards the outside, towards the watery chyme. And so what these bile salts allow us to do is they will form around the lipid droplets and break them down into smaller and smaller and smaller lipid droplets. And this will increase the surface area so that those lipases can actually access these smaller lipid droplets to digest the lipids. So the bile salts are really emulsifying the fat and helping it mix more nicely into the watery environment of the chyme.